Well, thank you very much. So David told me he was really mad at me when I showed up, and he said that he was going to make me go after Josh Schiffman, and it would be after Boston won 2 games against Toronto. So with that in mind, what I want to do today is take you through our thoughts of using zebrafish to study Li-Fraumeni syndrome, and then also telling you a little bit about some of the new progress we're making towards growing human tumors in the zebrafish. So the work I'll tell you about today has been done by 3 talented postdocs, one of which is here, Madeline Hayes. She has a poster tomorrow. I believe it's poster 23, so please visit her if you have questions about the P53 story. Here we go. So my lab uses zebrafish as a discovery tool. The fish is really a great model because the fish are small. In fact, they are 1 million times smaller than an elephant in terms of mass, which means that we can grow lots of these fish. In fact, my lab has 25,000 animals at any given time that we can study. So the fish are also great for in vivo imaging, which means you can look right inside them and actually image many of the hallmarks of cancer, which I'll show you briefly today. And importantly, we can also use the fish genetically, so we can knock out P53 in the zebrafish, for example. And then in beautiful work that Jason Berman is going to tell us about, you can also use the fish in chemical screening approaches to be able to identify new drugs that way. But of course, none of this really matters if the fish really doesn't recapitulate at least some of the molecular underpinnings of human tumors. And in fact, that's exactly what we see. Despite 450 million years of evolution, they are remarkably similar to what we see in human and mouse, for example. I'll get a handle on this in a minute here. So my lab really focuses on one tumor type, which is called embryonal Rhabdomyosarcoma. It's a common pediatric malignancy of muscle, and we know that P53 is deregulated in about 30% of these patients. And in fact, we also know that Li-Fraumeni syndrome patients will go on, or a subset of them will go on to develop embryonal Rhabdomyosarcoma. We also know that in Li-Fraumeni patients and in the patients that develop this de novo, local invasion and metastasis really are the major clinical problems, but we don't know what are the pathways that are driving that. So again, my lab uses zebrafish. I won't go through all the details, but we've developed a very nice model of embryonal Rhabdomyosarcoma. And in fact, we've discovered that, in fact, using this model and comparing it back to human tumors, that RAS is the dominant oncogenic driver in this disease, a really important finding when we start to think about what will synergize with this pathway. So in thinking about the role of P53 in this tumor, we undertook strategies to delete P53 in the zebrafish. So one of the reasons that we did this is that in normal tumors that will develop, usually you'll get a point mutation in P53 and a deletion in the other allele. So we decided that we would work, because Dr. Lane and Tom Look have beautiful point mutations, and Jason as well, that we would focus on this effort. And of course, what we can see, again, when Madeline made these mutants along with Myron in my lab, you could see that they nicely get rid of the P53 protein. And importantly, when you do basic assays like are they radiation sensitive or insensitive, you can see that the deletions, if they turn red, they're responding to the radiation. You can see that they really don't respond at all, indicating that the P53 response has been ablated. So again, why would we want to do this? Again, Dr. Lane nicely showed us that those point mutant zebrafish that he studies get one type of tumor, which is malignant peripheral nerve sheath tumors. But we know that Li-Fraumeni patients get lots and lots of tumor types. And remarkably, these fish that are full deletions in P53 actually get several types of tumors. They get these germ cell tumors at very low frequency, and then these other tumors at about a third of the animals, including angiosarcoma, a leukemia, which I'll tell you a little bit more about. And again, they also get these malignant peripheral nerve sheath tumors and a smattering of other types of sarcomas. So one of the other nice things about this model now is that we made these in a background that they are genetically identical, which means that you can take a tumor from one animal and implant it into another, and there'll be no immune rejection. And so this allows us to expand the tumors to be thinking about pathways like looking at drug sensitivity and the like. So, of course, what I told you too is we get these very strange looking leukemias in these animals, which we really had no idea what they were. And in fact, we then used molecular profiling, RNA sequencing, to ask the simple question, what are these tumors? And in fact, we looked at the angiosarcomas, leukemias, and other types of tumors, and we could see nicely that they identify a gene signature. But sadly, when we ask, is that gene signature found in human conditions, it was really nice that we could see that, in fact, they identify things that are involved in the immune system, but didn't identify the specific cell type that was affected within these tumors. So luckily for my laboratory and Madeline, we had actually mapped out the whole blood system in the zebrafish using single-cell RNA sequencing. And with this now in place, we were able to take that gene signature and splay it back onto those different populations. And we were quite amazed when we saw this, that in fact, they are NK-like or NK cell leukemias. In fact, they're an aggressive NK cell leukemia that is a very rare subtype of the human condition. And in fact, what was really remarkable is that when you look in the literature, p53 mutations have been reported sporadically in these tumors, yet it hasn't been linked as a causative agent. And in fact, the thing that's really exciting for us is that although this is a rare tumor, this is the first animal model of this disease, which really opens up a lot of possibilities for us. Okay, so again, I already told you that I'm really interested in rhabdomyosarcoma. So now what we did is we made rhabdomyosarcomas in the p53 deficient background and asked the simple question, what are they in fact doing? And I won't bore you with the details on what they're not doing, but what I will say is that we can see that, in fact, p53 can lead to tumors that are non-metastatic. We've injected these again into animals that are genetically identical at the site of the arrows here. And you can see that some of them don't metastasize. And yet others will metastasize quite readily when you put them into either the immune deficient background or the syngeneic animals. So when we quantitate this, it was quite remarkable that this is large transplant experiments, 160 animals, but you can clearly see that when you have p53 deletions, you in fact lead to highly metastatic tumors, or more predisposition to getting metastatic tumors, I should say. And in fact, my lab is well known for studying cancer stem cells. We looked at these, and in fact, remarkably, p53 deletion had very little to do with the overall population of cancer stem cells. So this left us really thinking about what to do next. And Madeline had this brilliant idea and worked with a student in my lab. She thought, wouldn't it be great if we could now serially transplant these tumors and get them to evolve and change to become more metastatic over time? And sure enough, that in fact works very, very nicely. You can see that as you serially passage these, again, they'll acquire new mutations. You can see that they become more aggressive over time and actually become more metastatic. And the real power of this approach now is that Madeline, again with another fellow now in the laboratory, is able to go in and use RNA sequencing to compare these tumors and has beautiful new pathways, some of which had already been studied in our lab, including Rho-A and plexin A1 as being drivers of this tumor. So hopefully in 2 years, we'll be able to tell you a little bit more about the specific pathways she's identified using this approach. So, of course, my lab commonly will make findings in the zebrafish and then translate them into mouse xenografts, cell culture, and patient tissues. Sadly, our work in Li-Fraumeni is sort of at that infant stage where we're really looking at the zebrafish experiments. But I wanted to divulge in the last 5 minutes and tell you about our new work in thinking about applying zebrafish to discover new drugs in rhabdomyosarcoma. So right now, if you want to take a drug cocktail into the clinic, you necessarily go through a mouse xenograft experiment. So these are great for preclinical drug testing. These animals have some limited capacity for imaging. They are expensive and they're not very high throughput. So it got us thinking, could we make a zebrafish that actually had no immune system? So that means you could take those tumors and implant them into the fish, and in fact, they wouldn't regress, but they would continue to grow. Maybe they would acquire a mutation, and you'd be able to image that over time. The fish allow for single-cell imaging. They're quite inexpensive. In fact, it's about 2 cents a day to raise a zebrafish, compared to about $1 a day for a mouse. And large studies and large-scale experiments are possible. So with these models now in hand, we've actually made immune-deficient zebrafish, and we can engraft a wide array of zebrafish tumors back into them. So this is just proof of concept, but we can image many, many things, including neovascularization, the dynamics of how cells can compete against each other within a tumor, ultimately to allow the most aggressive cells to grow. We can watch metastasis. For example, here you can see that a tumor that had been implanted into the eye now has migrated to the liver. And then you can also do really exciting things like looking in the tail, for example, and watching a single micrometastatic lesion land and then coming back days later and imaging that, in fact, it went on to form a micrometastatic or metastatic tumor. And this is just to remind me to tell you that this doesn't just work in rhabdomyosarcoma, but these are examples across many types of tumors that have been worked up in the zebrafish. Okay, so why would we want to do that? Well, my laboratory, again, wants to use zebrafish as a discovery tool of human cancer. So now the idea is that, well, what if we could make a zebrafish that would swim around in a 37-degree jacuzzi? Fish only live at 28 degrees, so it's quite amazing, but you can actually raise them in these jacuzzis and then implant them with a human tumor. And we can implant them into two areas. We can implant it into the eye or into the peritoneal cavity. And amazingly, with all of this, the fish can survive out to 45 days. So again, this doesn't really matter if the fish is not at least as good as a mouse. And so we've done the comparisons where we can engraft a rhabdomyosarcoma into a fish. These are human tumors that have been engineered to express a green fluorescent protein. And you can see and compare them to what grows in a mouse. Amazingly, they proliferate the same, they have very similar histology, and they express the clinical diagnostic markers or muscle markers that are similar to both humans and the tumors grown in the mouse. And so there are no differences between the models at all based on these experiments. I'm not showing you the other three examples that hold the same. But again, the power of this now is we can implant them into two places, either the eye or peritoneal cavity, so we'll get back to that in a few minutes. So why would we want to do this? Well, we want to be able to use the fish to treat them with drugs. So here's an example where we've now taken a DNA damaging agent along with a PARP inhibitor and complexed them together to ask the question, can they actually go and kill off human tumors grown in the zebrafish? And the reason we picked this is that this clinical combination has already passed phase 1 clinical trials, which really are the trials in kids that are really tough because that establishes whether the drugs are toxic to patients. And in fact, now we knew that based on these results and other results, which I'll show here, that in 4 separate examples of human tumors, again grown in the zebrafish, we could actually see that PARP inhibitors along with temozolomide had pretty nice responses in ablating the tumors. I didn't point this out, but these fish are orally gavaged, where we actually deliver the drugs at clinically relevant dosing and schedule into the fish. So one of the other powers of the fish, as I've shown you, is that you can image. So what we've now done is we've engineered human tumors to express this crazy set of proteins where there are 4 fluorescent proteins that are expressed in the cell cycle. And what you can do now is at single cell resolution ask, what are the cell cycle states of the individual cells? And just like I had shown you, the same cell cycle kinetics are found in mice and in zebrafish, and this is just quantified above. So why would we want to do this? Well, it's really difficult to tell if a drug combination is affecting cells at that early stage that you deliver them. And so now what we've been able to do is we've been able to complex our drug combination shown on the bottom and compare it to single agents or controls. And what we can clearly see, and it's a little bit of a busy slide, but we can image this over many, many days and serially image these animals. And I'll just blow this up so you can actually see what we're talking about. But you can see that when you have the potent drug combination together, they lead to a G2 cell cycle arrest that is much quicker than either of these drugs alone. And we think that that's the reason that they're going on to apoptose. And this is really very exciting because I believe this is the first time that we've actually been able to watch in real time a clinically relevant dosing schedule of any patient or any human tumor grown in vivo. So with that, I'll sort of end, but I'll tell you that this doesn't just work in these rhabdomyosarcomas. It works across multiple tumor types, including melanomas, triple-negative breast cancers, a wide range of sarcomas, renal cell carcinoma, CML. And of course, the real goal here is that we could actually take a tumor from a patient and engraft it into zebrafish, which sounds like science fiction to me. But what we did is we paired up with Dan Haber's group at the Cancer Center, who could actually get circulating tumor cells from patients. There are very rare cells that are shed from your tumor that go into your blood. He was able to culture those ex vivo for a few days and then give them to us and grow them into mice or into fish. And what was really remarkable is that, in fact, human patient-derived xenografts can now grow in our zebrafish using various models. So looking to the future, our dream is that, in fact, we'll now be able to take these and start to use them in sort of preclinical drug development like I showed you, but the real dream is that we'll have patients that come into Mass General, we'll implant their tumors into an avatar army of zebrafish, and then we'll treat them with open clinical trial drugs, and we'll help stratify those patients that respond in their avatars and report that information back to the clinic. We think that this could really change the way that we think about clinical trial design because, for example, this may not be a Mass General drug, but it might be a Dana-Farber drug, which is just across town, and we can tell that patient to go across town to get their therapies rather than sealing their fate by just walking through the doors at Mass General. So with that, I'll thank my lab. Again, Madeline Hayes and Myron really spearheaded the P53 story, a real tour de force. And then Yan Chawan really worked up the immune deficient zebrafish project. Thank you.
Thanks. Arnie again.
Sorry to be so loquacious here. First of all, that was absolutely terrific.
Thank you.
I only must have missed something. I know that fish have an adaptive immune response, and they're not rejecting the human tumors. So why is that?
Okay, I'm sorry, I took my slide out, but we've made these fish, again, I'm sorry, they're nod-skid, IL-2 gamma null complement as what we would see in the fish. I'm sorry that I took out the creation of the lines, but yeah. Josh, last question? Yeah.
Great talk. I love the avatar piece of it.
Thanks.
Will this work with IV medication, or is it only medicine that's orally gavaged?
We haven't tested any of the IV. I think that if we did that, we would probably do peritoneal type of injections. It's hard to sort of access like a tail vein in the fish. But if you had asked me a few years ago, could we raise a fish at 37 degrees, have it swim around, implant a human tumor, and then orally gavage a clinically relevant drug dosing, I think probably it's an addressable question. But it's amazing. So, anyway, thanks.